Welcome to our poster on a randomized double-blind evaluation of CRS3123 versus oral vancomycin in adult patients with C. diff infection, or CDI, which is a phase 2 clinical study currently enrolling patients at sites in the US and in Canada. This work is funded entirely by a contract from the National Institute of Allergy and Infectious Diseases at NIH. I'm Urs Ochsner, co-founder and CEO at Crestone, a small biopharmaceutical company in Boulder, Colorado. I will give you an introduction to the CRS3123 program. CRS3123 has unique properties that make it an optimal agent for CDI treatment. It is a fully synthetic small molecule drug and has excellent potency against C. diff, including hypervirulent strains with an MIC90 of 1 microgram per ml. Importantly, CRS3123 has a very narrow spectrum of activity targeting C. diff with unprecedented specificity. It inhibits methynyl tRNA synthetase, or MEDRS, which is essential for protein synthesis in C. diff. Therefore, it inhibits not only growth of the pathogen, but also immediately stops the production of the toxins that cause the disease, and also blocks the formation of spores that are responsible for recurrence. Since this is a novel mode of action, there's no pre-existing resistance. Compared to other C. diff drugs, CRS3123 has comparable activity against C. diff, but is not active against almost all beneficial bacteria in the gut, as shown by in vitro testing and in a microbiome study conducted in a phase 1 clinical trial. The narrow spectrum of our drug is due to the phylogeny of the MEDRS target, which exists in two types. Only bacteria with the type 1 enzyme are susceptible, which includes C. diff. In contrast, bacteria with the type 2 enzyme are not affected by CRS3123, and this includes all gram-negative bacteria such as Bacteroides, Prevotella, Actinomyces, and Bifidobacteria, and many others that are part of a healthy gut microbiota. CRS3123 has shown excellent efficacy in the hamster model of CDI, with most animals surviving through the end of the study. In contrast, most vancomycin-treated hamsters died 10 days after treatment ended, likely due to recurrence. We hope these promising data to translate into low recurrence rates in CDI patients. CRS3123 was found to be safe and well tolerated in phase 1 clinical trials conducted in healthy participants. The single ascending dose study had 5 cohorts with single doses ranging from 100 to 1200 milligrams. The multiple ascending dose study had 3 cohorts receiving 200, 400, and 600 milligrams of the drug twice daily for 10 days. Most adverse events were mild, and there were no serious adverse events. The drug had a low impact on the gut microbiome that confirmed the high selectivity despite high concentrations in the gut. Definitely a favorable drug profile of CRS3123 to warrant its progression to phase 2. The microbiome study conducted during phase 1 showed very modest changes in the healthy gut microbiota during treatment. Importantly, no major phyla were lost, and the composition of the gut microbiota was restored to baseline levels within a few weeks after the end of treatment. For the phase 2 study, we chose regimens of 200 milligrams and 400 milligrams twice daily for 10 days. And with that, I'm happy to introduce Dr. Katie Johnson, Crestone's Director of Clinical Research, to provide an overview of the ongoing phase 2 study.
Thank you, Dr. Ochsner. I will be presenting a brief summary of our ongoing phase 2 clinical trial. We are studying adult patients with either primary or first recurrence of C. difficile infection. Patients included in this trial must have a positive GDH and toxin A or B test using the Abbott Alere Quick Check Complete. The key exclusion criteria for this trial are those patients with inflammatory bowel disease, patients with recent C. diff infection that did not respond to vancomycin, patients with underlying immunodeficiency, and patients with severe hepatic impairment, such as cirrhosis or end-stage liver disease. Patients will be randomized to one of 3 treatment arms. There are 2 treatment arms with our selected dosages of CRS3123 and a treatment arm of standard course vancomycin, 125 milligram 4 times daily. The treatment course will be 10 days. We plan to have 30 clinical sites and approximately 90 to 108 patients enrolled. These are our current clinical sites in the United States and Canada. The listed investigators on the right-hand side of the slide are currently enrolling subjects. This is a schematic of our study design. We have a screening window of 24 hours, followed by a 10-day treatment course with 3 in-person visits. Then there is a 60-day follow-up period with 4 in-person visits. Importantly, we will be monitoring for CDI recurrence during this time. Our test of cure visit is 2 days after the end of treatment visit or at day 12. In our trial, we will be assessing the efficacy of CRS190021 in patients with CDI. We will be doing this by measuring clinical cure of C. diff disease after a 10-day treatment course, but also by the lack of recurrence of CDI during the follow-up period. We are also interested in the safety of our treatment in those patients suffering from C. difficile infection. Lastly, we want to gain knowledge of how patients feel when they take our drug while they have C. diff infection. In summary, CRS190021 has several key attributes that make it an ideal drug for treatment of C. difficile infection. It is the narrowest spectrum of all available treatments with selective inhibition of C. difficile. This is similar to precision medicine for C. diff infection. This selective inhibition will lead to a faster recovery of the gut microbiota. CRS190021 inhibits toxin production, leading to a decrease in severity of illness. Importantly, CRS190021 inhibits spore formation, which will lead to lower CDI recurrence. Why do we need another C. diff therapy? It is because this treatment will improve patient outcomes. It gives clinicians another treatment option in their armamentarium. This treatment could improve the health economics of CDI by decreasing hospital length of stay with fewer hospital readmissions and fewer outpatient clinic visits. Thank you for your time.